Welcome to this explainer on the Cambridge AS and A-Level Biology 9700 exam strategy. Look, today we're not just, reviewing basic biological facts. We are literally decoding the examiner's playbook. We've all been there, right? You know the material backward and forward, but you still inexplicably drop marks on the actual test. It's beyond frustrating. Well, you are definitely in the right place. We're going to transform how you approach this high-stakes exam using a highly tactical approach. Get ready to see the syllabus through the exact eyes of the person grading your paper.
Okay, let's dive right into this. I want you to watch carefully here, because honestly, 9 out of 10 students get this exact type of question wrong, and it quietly destroys their grade. On the left side of this slide, we have a classic flawed answer: transmission occurs through contact. I mean, it sounds true, right? But the examiner's red pen is already out. No marks. Now, shift your focus to the top band answer on the right. Transmission of HIV occurs via direct contact with bodily fluids. See the difference? The generic answer on the left completely misses the context and the specific terminology required. Let me show you exactly where this error creeps in, so you can lock in those marks instead. Imagine we're looking at a huge classroom projection of this Golgi body. Topic 1.2 on cell structure is an absolute heavy hitter. It accounts for roughly 15% of your paper 2 marks. But here is the massive trap. Students memorize a generic, high school level definition like the Golgi processes proteins, and that superficial knowledge just bleeds easy marks. Notice how the Cambridge framework prioritizes analytical depth. A top band answer doesn't just name the organelle. It links the structure directly to its function, noting very specific things like adding carbohydrate chains to form glycoproteins, which is driven by the specific data in the prompt.
So, what does this all mean? How do we systematically build that top brand answer every single time? Well, you need the 4-step answer framework. It's like a guaranteed recipe. First up, you identify the core biological concept. Second, you extract specific evidence or data from the provided tables. Third, apply scientific reasoning to explain the causal relationship. And finally, and this is crucial, wrap the whole shebang up in the precise examiner wording expected by the mark scheme. This framework is the bridge that takes you from basic rote memorization to complete exam mastery. And this brilliantly illustrates exactly how we apply that 4-step framework to a topic like active transport. Focus on that examiner-approved top-band response on the right side of this full-screen image. Look at how it starts with syllabus-precise terminology, extracts specific data points and unit citations, bridges the concept to the evidence using logical reasoning, and rigorously follows command words. Now compare that to the flawed answer on the left, which, let's be honest, is just unstructured fluff. The framework literally forces you to be specific.
That specificity becomes incredibly important when we look at terminology. A massive pitfall for students, a real grade killer, is using confused, generalized vocabulary. For example, casually throwing around the word denaturation when the question is actually asking about the specific mechanism of inhibition. Look, examiners are trained to look for precision like a hawk. If you use a generic descriptive word where a specific analytical mechanism is required, you lose the mark instantly. Absolutely instantly, even if you completely understand the broader concept. Let's apply this to enzyme activity versus pH. If you look at the bell curve on screen, the flawed student answer completely ignores the specific values on the graph and uses descriptive language without evaluating the evidence at all. But look at the improved top band answer on the right. It correctly uses the term denaturation, specifically in the context of enzyme activity above the optimum pH. It integrates accurate units and data interpretation directly from the bell curve, evaluating the quality of evidence. That is exactly what they want to see.
So the absolutely crucial point here is avoiding what examiners actually call the top 3 mark killers. As we look at the microscopic mechanics of enzyme inhibition here, note the major red flags. Number 1, missing units in your calculations. Number 2, using non-specific terminology. And number 3, failing to relate your answer to the provided experimental context. To put that into perspective, if a question gives you inhibitor concentration from 0.1 to 0.5 micromoles per decimeter cubed, your answer absolutely must use those exact figures to prove your analytical depth.
To prevent those mark killers before you even put pen to paper, you need a secret weapon. We call it the deconstruct, solve, evaluate loop. You must deconstruct the prompt to identify exactly what is being asked. Solve the problem by selecting the right analytical framework, and then, don't skip this, critically evaluate your final answer to ensure it actually aligns with biological principles. Remember, marks are awarded for your procedural framework and your scientific reasoning process, not just for randomly dropping facts onto the page.
This procedural rigor is totally non-negotiable in Topic 6.2 on post-transcriptional modification in statistics. Just scan this statistical tool selection chart. Exam mastery requires you to know exactly when to pull which tool from your toolbox. Are you comparing the means of two sets of data? You're going to use a t-test. Are you testing the significance of differences between observed and expected categorical data? That's chi-squared. Looking for a relationship between variables? Use correlation. Making the right choice here is step 1 of the deconstructive phase.
Let's see how this builds into the method mark strategy, using the mass flow hypothesis in phloem as our visual example here. Notice the clear working out section on the bottom right. The examiner alert here is crystal clear. Students lose marks by not showing their working. It happens constantly. You must break your analysis into logical steps and literally write them down. Even if you make a minor calculation error and your final result is totally wrong, showing your clear, logical process can secure you vital method marks that literally save your grade.
Another major differentiator between an average grade and an A-star is context. Ordinary revision often leaves you with just isolated facts, but high-achieving candidates do something entirely different. They integrate their knowledge across different syllabus areas. You literally have to link your brain to the specific botanical context provided, just like this graphic shows. An answer resting on generic context will always score lower than one that is heavily contextually linked to the stimulus material. You have to prove to the examiner that you understand how these biological concepts behave in the specific, real-world scenario they've handed you.
Let's revisit our cold open mistake by examining this full-screen breakdown of disease transmission. Notice how the top band response completely relies on contextual links. It doesn't just vaguely say transmission occurs. Nope. Instead, it identifies the causative agent, it links it to specific bodily fluids, and it incorporates precise figures like a 10% increase in risk. It connects the evidence to the concept using logical causal chains. This right here is what it means to write with analytical depth rather than just superficial breadth.
Which brings us to another notoriously complex syllabus target where students frequently stumble: the respiratory quotient, or RQ calculation. It's a perfect storm of data interpretation, formula execution, and biological evaluation. Students often fail here, not because they don't know the formula, they usually do, but because they fail to execute the correct notation, and crucially, they completely forget to perform comparative analysis. Now, what's really interesting about this slide is the red pen warning right in the middle. Mistake 3, evaluation failure. High-mark candidates are literally required to compare approaches and justify their conclusions. When you calculate an RQ value, you can't just leave the number hanging out there on its own. You have to compare it, identify similarities and differences, and evaluate what that actually means for the organism's respiration in that specific given context. All right, let's pause for a second for a rapid-fire interactive challenge pulled straight from the Principal Examiner reports on Topic 18.2, Assessing Biodiversity. Imagine you see this calculation on a candidate's paper. 50 minus 20 equals 30. Mathematically, it's flawless, right? But in the context of the Biology 9700 exam, it is an absolute mark killer. You have exactly 5 seconds to spot the missing element. What is wrong here?
Missing units. Yep, if you didn't say units, you just dropped an incredibly easy mark. Whether you are calculating species abundance, genetic variation, or ecosystem biodiversity, dropping your units, like individuals per unit area, is the absolute fastest way to leak marks. Examiners note this exact mistake year after year. A number without a biological unit is just a number. It is definitely not scientific evidence.
Which brings us to the ultimate exam hack, pulled directly from Cambridge Principal Examiner reports. Quote, students lose marks by not showing working. Even if your final answer is wrong, method marks can save you. Seriously, stop doing the math in your head. Stop rushing to get to the final number. The exam is testing your logical reasoning process just as much as your final conclusion. Show your steps, every single one.
Let's lock in everything we've covered today with the A-Star Mastery Checklist. Number 1. Deconstruct the prompt before you write a single word. 2. Extract specific data and cite it directly. 3. Use precise, syllabus-specific terminology. Absolutely no generic fluff allowed. 4. Show your method clearly to secure those procedural marks. And 5. Always evaluate the reasonableness of your answer within the given biological context. You now have the ultimate blueprint for structuring an elite top-band answer. But there is one final piece of the puzzle that controls exactly how you apply this framework. You've mastered the 4 steps, but do you know the secret language of examiner command words?
Like, what is the actual structural difference between a question that asks you to describe versus one that asks you to explain? Understanding that subtle difference is the ultimate key to unlocking the highest grade boundaries. Think about that, and I really look forward to revealing the answer in our next explainer. Keep practicing your past papers and stay incredibly focused.